Okay, think about this for a second. Without a massive, invisible army of tiny workers inside you, you literally couldn't digest your food or even process your next thought. Today, we are pulling back the curtain on these unsung heroes of our bodies, enzymes. They are the secret powerhouses that make life itself possible.
I mean, have you ever really thought about it? You eat a burger or a salad or anything really, and somehow your body magically turns that into fuel. Fuel for running, for thinking, for breathing. The secret ingredient in that whole incredible process? You guessed it, enzymes. So, here's our game plan. First up, we're going to properly meet these things called catalysts. Then, we'll dive into their secret handshake, known as the lock and key model. After that, we'll watch them do their day job, breaking down our food, and figure out what conditions they need to work best. And finally, we'll pop into the lab to see how scientists actually test all this stuff.
All right, let's start at square one. We hear the word enzyme thrown around all the time, right? Especially when we're talking about digestion. But what in the world is an enzyme?
Okay, let's unpack that definition. First off, enzymes are proteins, but they're special proteins. They're biological catalysts. Just think of a catalyst as a super efficient matchmaker that speeds up a chemical reaction like crazy, but doesn't get used up in the process. It can just do its job over and over and over again. And when I say speed up, I mean it. Some of the reactions they handle would take years to happen on their own. With an enzyme, we're talking seconds. Without them, all the chemical reactions that keep you alive, digesting your lunch, building new cells, would be so slow. Life as we know it just wouldn't be possible. So what's the secret? How does an enzyme know exactly which reaction to work on out of the thousands happening in your body all the time? Well, the answer is actually pretty brilliant, and it's called the lock and key model. So the idea here is that an enzyme is shaped like a very specific lock. It has this one little area on it called the active site. And only one particular molecule, which we call the substrate, has the perfect shape to be the key that fits into that lock. It's a super specific fit that makes enzymes so precise. So let's watch it happen. The substrate, our key, finds the enzyme's active site, our lock. Click. They bind together, a perfect fit. Then, the enzyme works its magic, either snapping the substrate into smaller pieces or joining it with another. Then, it releases the new products, completely unchanged and ready for the next customer. It's just this incredibly efficient, non-stop cycle. But what happens if you bend the lock? You know when you cook an egg and the clear part turns solid and white? That's called denaturation. The heat permanently changes the shape of the protein. Well, the same thing can happen to an enzyme. If its active site changes shape, the key, the substrate, just won't fit anymore. And if there's no fit, there's no reaction. Simple as that. And this brings up a really important little detail. You'll notice we say an enzyme is denatured, not killed. That's a super common mistake, but the distinction matters. Enzymes are just complex protein molecules. They were never actually living things to begin with.
Okay, enough with the theory. Let's see these little superheroes in action in a process that's happening inside all of us right now, digestion.
See, the food we eat is made of these massive molecules. Carbohydrates, proteins, fats, they are way too big to pass through the walls of our intestines and into our bloodstream. It's like trying to get a bus through a regular-sized door. So that's where our digestive enzymes come in. They act like molecular scissors, snipping these giant molecules into tiny pieces our body can actually absorb. And it's a very organized demolition job. You've got specific enzymes for each major food group. An enzyme called carbohydrase breaks down big carbs into simple sugars. Protease takes care of proteins, breaking them into amino acids. And lipase tackles fats, splitting them into fatty acids and glycerol. Each one has its own specific target. So we know enzymes are picky about what they work on, but it turns out they're also super picky about their work environment. You can think of them as the Goldilocks of the molecular world. Exactly. For an enzyme to do its best work, things can't be too hot or too cold. The pH can't be too acidic or too alkaline. Everything has to be, you guessed it, just right. If the conditions are off, the enzyme slows down or it might just stop working altogether. Let's look at temperature. As things warm up, molecules move faster, so the reaction speeds up and up until it hits this perfect peak. That's the enzyme's happy place, its optimum temperature. But if you keep cranking up the heat, that delicate protein structure starts to fall apart. The active site changes shape, the enzyme denatures, and the activity just plummets straight down to zero. And it's pretty much the same story with pH, which is just a measure of how acidic or alkaline something is. Every enzyme has its own ideal pH where it works fastest. If you stray too far from that sweet spot in either direction, the active site gets warped, the enzyme denatures, and the whole reaction grinds to a halt. And this is where it gets really amazing. The enzyme in your stomach that breaks down protein, it loves a super acidic environment with an optimum pH around 2. That's like lemon juice acidic. But an enzyme in your small intestine is built for a totally different world. It prefers a more alkaline pH around 7.5. They are perfectly adapted to do their job exactly where they are.
Okay, one last factor. How much work is there to do? Well, the more substrate you add, the faster the reaction goes, right? More keys for the locks. But there's a limit. Imagine you have 10 workers. Those are your enzymes. If you give them 100 tasks, the substrate, they're all going to be busy. At that point, giving them another 100 tasks won't make them work any faster. They're saturated, and the reaction rate just flatline.
All right, so that's the theory. It all sounds great. But how do we know this is how it works? Well, scientists can put it to the test. And this experiment with an enzyme called amylase is a perfect example. So amylase is the enzyme that breaks down starch into sugar. You actually have it in your saliva. It's why a cracker starts to taste sweet if you just let it sit in your mouth. So scientists can use this reaction to figure out the exact pH where amylase works the fastest.
Here's the basic setup. They get a bunch of test tubes, and in each one they mix amylase, a starch solution, and a buffer solution to set a specific pH. They keep the temperature the same for all of them. Then, here's the clever part. They use iodine, which turns black if starch is present. So every 20 seconds, they test a drop. As soon as the iodine stops turning black, they know the starch is all gone. The whole point is to see which pH gets the job done the quickest.
And what did they find? The results were crystal clear. At a pH of 7, it took only 60 seconds for all the starch to disappear. At any other pH, whether more acidic or more alkaline, it took way longer. The experiment proves it. For amylase, the just right condition, its optimum, is a neutral pH of 7.
So there you have it. Enzymes are these incredibly specific, super powerful, and kind of sensitive little protein machines that run the show inside of us. They are the tiny engines that make everything happen. And that leaves us with one last massive question. Now that we understand how these biological engines work, what could we engineer them to do for us? Could we design enzymes to break down plastic waste in our oceans or to hunt down diseases in our bodies? The possibilities are pretty mind-blowing.